Have you ever wondered what it'd be like to live amongst dinosaurs?
Ever wondered how Earth would be if a species never went extinct? Or if a species came back?
That's what me and my partner Kaden will be exploring today. Today we'll be looking at the possibilities of bringing back extinct species.
The process of doing so, how Earth and the animal would adapt, and the ethics behind it all.
We were able to contact a professor from the University of British Columbia, and he was kind enough to let us interview Dave Ng. He was able to give us a butt ton of very useful information to help us. We will be showing clips of our interview with him throughout our video. As humans, we must acknowledge the fact that many of these species went extinct because of our actions. We need to understand that bringing them back won't undo our wrongs. With that being said, let's explore some species that are being brought back and the ones that were successfully brought back. CRISPR-Cas9 technology is a very effective way to recreate extinct species. CRISPR is gene editing software. With CRISPR, we are able to modify a specific part of the genetic code, and for that reason, CRISPR is a very viable option for recreating extinct species. A DNA-cutting protein called Cas9, which is what you see in blue, and a single RNA strand, which is in red, makes up the CRISPR-Cas9 complex. When the Cas9 complex finds the targeted area, it unwinds the double helix of the DNA and cuts out the code we don't want. After it makes the cut, the RNA strand is used to write a new code along with different enzymes to mutate a specific gene to change the genetic sequence in the DNA.
If we found just a few bone fragments of an extinct species, we could extract and analyze the genetic code to get a small portion of the genome. But this process could take an immense amount of time, and this does depend on the species, because the longer the species has been extinct, the harder it would be to find good samples of the DNA.
Thing you want to bring back, right? Like if it's a plant that's extinct but has only been extinct, I don't know, like 100 years or something like that, there may be samples, specimens in a museum where even though the plant is dead and there's no more, you may be able to extract like really decent amounts of DNA.
Let's just say that we were able to find a lot of good samples and could create a good layout of the genome. We could then line up the code of a similar living relative and use the CRISPR-Cas9 complex to genetically modify the relative until it matches the code of the extinct species, returning the code to its original state, would transform the current species back into its extinct ancestral species.
CRISPR has already been used to genetically modify a few species in the last few years. The Ecus quagga quagga, or better known as the quagga, was a donkey-zebra hybrid, or as we like to call it, a zonkey, and went extinct in the 1800s due to overpoaching. They lived mainly in Africa, and because of their unique hide and delicious meat, they were killed, until 3 remained in captivity and then none. Scientists were able to analyze and extract genetic code from the quagga's hide. With this information, they were able to genetically modify zebras' hides to look very similar because of their extremely similar genetic code, and were able to bring them back from extinction.
But let's say you do get progeny. Let's say you get a baby mammoth/elephant. So the idea there is that that hybrid would have some mammoth DNA and some elephant DNA, right?
Scientists from the journal Current Biology explored the concept of bringing back the Christmas Island rat. The scientists were able to retrieve almost all of its genetic code. The extinct animal is also very closely related to a living rat species, the Norway brown rat, which it shares 95% of its genome with. This made the Christmas Island rat an ideal test case, as the scientists could directly compare the two genomes and analyze the difference between the two. After finding the difference, they could identify distinct traits of the Christmas Island rats and know how to change the DNA of the Norway brown rat to match their extinct relatives. These are the reasons that CRISPR is one of the best and most modern ways that we could recreate an extinct species. Backbreeding is another way that we could recreate an extinct species, but it is unreliable and poses issues to the species itself. First, let's explain what backbreeding is. Backbreeding is the term used to describe the use of selective breeding to resurrect specific ancestral traits within populations of living species, meaning that we are trying to have a specific trait expressed in the current species that the extinct species had, eventually making them the same.
There are some requirements that make backbreeding an unreliable but possible way of recreating species. The desired trait must still persist in a living relative, meaning that backbreeding is only an option when the living relative is very genetically similar to the extinct species. There's also no guarantee that the selective trait will be expressed due to the genetic and environmental interactions with the living relative. The environment has changed over time, and the phenotype may not be expressed at all in the relative.
Another way to recreate extinct species is cloning, or somatic cell nuclear transfusion. Cloning is a process of recreating an exact genetic copy of an organism. Cloning is done by injecting the nucleus of an adult somatic cell into an egg cell, and then is reprogrammed by the host egg cell.
Cloning is very helpful for species that have gone extinct in recent years. For cloning to work, a somatic cell needs to be alive, so species that have been extinct for thousands of years aren't an option unless we are going to find a living cell.
The cell would then develop after the egg cell becomes fertilized by a sperm cell. The organism resulting from the cloning would have an identical genetic code and genome as the donor of the somatic cell.
Lots of new research is happening every day for cloning, and progress is happening. There have been many cases of successful clones with an exact match of a genetic code. A few successful clones have been several mice in 2008, a bucardo in 2003, and South Korea was able to create mammoth embryos in 2015. Cloning is a much more favorable option to backbreeding, and it is much more efficient and accurate. Those are the current ways that we're able to recreate a species at the moment.
A lot of it depends on your worldview around working with animals, I guess, right? The other ethical angle that I think is worth considering is what, like, why? Like, why are you doing this exactly?
Yeah.
Like, what's the point? Is there an applicable point? Microbes, for example, and maybe some really primitive plants where the things they are used to don't exist anymore, like sort of atmospheric compositions and stuff like that, right? So you can imagine that presumably they would have a tough time unless you are able to set up an environment where you can mimic the thing that it's able to handle, right?
So again, we can recreate extinct species in a few ways, but that doesn't mean we should. Lots of negative things could happen from doing this, and it might not be something that we benefit from.